So what I'll be talking about is just the basics of the microbiology, which we have, I should say, which we have forgotten, but which we all of us have gone through during our study period. But once we have completed, this is something which we haven't, we never attempt to review. But then this has become very important in today's world, because antimicrobial resistance, this has become a problem all over the world, and we are on a war path, I should say, to preserve our antimicrobials which we are using presently. So I'll be just telling the basics which we all have gone through earlier. So microorganisms, when we say microorganisms, what we understand is those organisms which are too small to be seen with the naked eye. And these are, we may not think that, because this is not visible to the eye, doesn't mean that it is not there. It is everywhere. It is just about everywhere. It is there on the tabletop, it is there on the chair, it is there in the environment, in the air, in the floor, in the water, almost everywhere. And they are such a unique, organism, in the sense that they have the capability to thrive in the extremes of temperature. The majority of these, they survive in temperate and where we feel comfortable. But then some of these microorganisms, they survive in the extremes of temperature, like even in the Antarctic. We have detected organisms, and even in hot springs, we have detected these the microbes. And even in those conditions where too high of salt concentration, too alkaline, too acidic, they have the capability to survive. So if you go, the gel to live is much stronger, and they have evolved in such a way that compared to human, we humans are nothing. They have a better way of living and surviving in this world. So if we go back to the history, as a microbiologist, as a medical professionals, we just can't neglect. We should always be, thankful to these people who led us to the discovery of the microbes, and many a theory that makes us realize that organisms are the causative agents of these infectious diseases, and many of their theories, many of their postulates, till today, it still holds true. So had it not been for Anthony van Leeuwenhoek, who discovered microscope, he was not a microbiologist. For that matter, he was not even a medical professional. He was not even a scientist. He was just a businessman who deals in gems, but his hobby was, handling the glasses. That is how he invented the microscope. When we talked of microbes, we know that there are bacteria, there are viruses, there are fungi, and there are parasites. So can be grouped as eukaryotes or the prokaryotes. So we, as I have mentioned, we are there. But then the number of the microbes is much more in our body. We tend to think that we are very much clean. There is no microbes, there is no organism in our body. But if we think, please remember that the number of cells which is present in our body, it's supposed to be very high, but then the number of microbes which is present in our body is much more higher than the number of cells present in our body. So when I say that it is everywhere, does it mean that they are always the cause of infectious disease? And is these microbes always responsible for diseases? No, it is not. Because let us remember that we have good microbes, we have bad microbes. Okay. Yes, again, our gut flora has more than 500 species of bacteria. Forget about the number of the bacteria. The number of the species number is almost more than 500. So when I say it is helpful, how it is helpful? So like basically in the normal situation we are in a rapport with the microbes. They contribute a lot in our health also, because they help, these microbes in our body, they help in digestion. They are responsible for synthesis of vitamin A, biotin, and etc. And whenever there is onslaught of the bad bacteria, pathogenic bacteria, they are there to defend us. Some of these, they act as a defense line. And then some of the bacteria, they are responsible for producing chemicals that can destroy the bad bacteria. Like, for example, penicillin. Okay. Penicillin, originally it was from the fungus Penicillium. So they produce enzymes, chemicals, which can act on other bacteria which are responsible for causing disease in our body. So that means, basically speaking, these microbes, they, on the other hand, they are helpful. They provide, they are part of the good immune system in our body. Whereas on the other far spectrum, they are responsible for causing invasive diseases. Okay. Yeah. So what is needed is a balance. And what we are concerned as a medical professional is those diseases. It may be a simple soft tissue infection, or it may be an invasive disease that we are concerned about. So though I have said that there are classes of microbes, what we'll be dealing today, what we are really concerned with the antimicrobial stewardship program is a group of that microbes, that is bacteria and the antibacterial agents. Because drug resistance, though drug resistance is there in case of viral diseases, fungal diseases, and also in parasitic diseases, it is more the issue and the problem that we are facing today is mostly with the antibacterial agents. So bacteria, as we know, it has got various shapes and size. It may be round, which we term as cocci, and it may be, rods, or that we call it bacilli. And then on the other hand, we have spiral-like bacteria that we call as spirals. And some of these, they are spore forming, some do not form spore. So then again, we know that depending on a gram stain, we classify the bacteria into gram-positive bacteria and the gram-negative bacteria. So this can be, depending on the shape, it can be gram-positive cocci or gram-negative cocci, or it can be gram-negative bacilli or gram-negative cocci. So what makes it gram-positive? What makes it gram-negative? During the gram staining, it is because of the difference in the cell wall structure of the cell wall structure of this bacteria. Because so what comes into our mind? This is something which we come across every day. So among the gram-positive cocci, we have Staph aureus, Streptococcus species. And when we talk of gram-negative bacteria, we think of E. coli, Pseudomonas, Salmonella, Shigella, and Klebsiella pneumoniae. So the typical structure of a bacteria, this has got, this is an eukaryote, it's a prokaryote, and so this has got a cell enveloped. This is a cell wall, and this cell wall, this is responsible for maintaining the rigidity and the shape of the bacteria. And this is separated from the cytoplasm by a cytoplasmic membrane. Okay. And in this cell wall structure, okay, that will come. Then this cytoplasmic membrane, it has got ribosomes, then it has got granules, volutin granules, then it has got vacuoles, all those things. And then the nuclear material in case of this bacteria, it doesn't have a very, it doesn't have a proper nucleus. What we have is a coil of DNA material, okay, which does not have a membrane, and this a term as nucleoids. That is why we said that is the characteristic of prokaryotes. So this is it's the cell membrane that divides the cytoplasm and the cell enveloped. And when we talked of the cell wall, it has got a very important role. As I have mentioned, this is responsible for the shape and the size and cell rigidity. And so in addition to that, besides this, we may have structures like capsules, flagella and fimbria. Some organisms may or may not, some may have flagella, some may not have flagella. Some may have capsules, some doesn't have capsules. And those organisms that have capsules, they are much more, they contribute to the strength of the bacteria, and many a times they contribute to the resistant mechanism as well. So in the gram-positive, it is because of this difference in the cell wall structure of the gram-positive organism and the gram-negative organism that makes a bacteria gram-positive or gram-negative on gram staining. So in case of gram-positive bacteria, what we have is a thick peptidoglycan layer, whereas it is much smaller. Like in case of gram-positive bacteria, it contributes about 90%, whereas it's only about 10 to 20% in case of gram-negative bacteria. And this has got a periplasmic space. Like this peptidoglycan is there in the periplasmic space, which is not so in case of gram-positive bacteria. And this gram-negative bacteria have an outer membrane protein. So, coming to the antibiotics, there are different groups of antibiotics that we know. So, but basically, at the basic level, what they do is, we classify this antibiotic as those antibiotics which act on the cell wall synthesis. When they disturb the cell wall synthesis, they destroy and then lead to the destruction of the bacteria. So, for example, the penicillin groups, then cephalosporins, carbapenems, monobactams, and then again, of course, we have vancomycin and bacitracin. They all act on cell wall synthesis. What they do is, there are different, depending on the group, there are different strategy. They try to mimic a portion of the peptidoglycan. A chain of the peptide in the peptide, they try to mimic this antibacterial, they will mimic it, this antibiotic. So the normal process of peptidoglycan formation is going on in a live bacteria. But what happens is, when the bacteria attacks this, when the antibiotic attacks this bacteria, a portion of the antibiotic will mimic a portion of the cell wall synthesis of peptide chain. So they will take up that portion. They will miss, and then that will not be available for the cell wall synthesis. So that is how it gets degraded. And of course, there are other mechanisms as well. It may form a larger molecule so that cell wall synthesis is hampered. So basically, it is the inhibition and the destruction of the cell wall synthesis. Then another group is the the one that acts on the cell membrane is the polymyxins. They will cause hydrolysis, and that will lead to cell membrane destruction. Then there are a group of antibiotics again that will interfere in the protein synthesis in the ribosomes. So in the protein synthesis, there are there are again two strategies, or rather there are other strategies as well, but the important one is, some of these, they will target the 30S subunit of the ribosomes, and another will target the 50S ribosomes. So this is inside the cell. These ribosomes are there inside the cells, as I've shown in the earlier figure. So what happens is, suppose if it is an aminoglycoside, it will need to go. It acts on the 30S ribosomes. So they need to enter the cells inside. They have to pass through the cell wall, they have to pass through the cell membrane and go inside so as to act on that ribosome, 30S ribosome unit. But then, what happens? So they just can't enter. So they need a pump system to traverse this cell wall. So this is again oxygen dependent process. The entry to the cell wall is oxygen dependent process, and then only in presence of this oxygen dependent process they will enter the cytoplasm and act on the ribosomes. So that is why we say that aminoglycosides do not act on anaerobic organisms. Why so? Because there is a component for the drug to act on these ribosomes. There is a component that require or that is oxygen dependent. So in the anaerobic condition, that will not be available. So it doesn't work. Again, those which act on the 50S subunit includes like macrolides, linezolates, chloramphenicol, streptogramins, all this. Then another group is that inhibits nucleic acid synthesis, like folate synthesis, and then again DNA quinolones, they act on the DNA gyrase, and then so RNA polymerase disturbance like rifampicin. So we always say that we always tend to give sulfonamides and trimethoprims together. Though both of these drugs act on the bacteria through the same pathway, that is folate synthesis, again, though it is the same pathway, it is in different steps. That is why, and they have a synergistic effect. That is why we give it this antibiotic together. So how does it become resistance? How does the bacteria become resistant? As I have said earlier, these microbes are million times much more smarter than we human beings. They have a way of evolving. Their evolvement is so smart that sorry, they will try to change the antibiotic. They will, when the antibiotic is there, they will try to change some molecular structure or some chemical nature of the antibiotic so that it doesn't attack them. Then again, another is they will bypass the effect of the antibiotics. And then there are many times where they will change the target of the antibiotics. Instead of the target which is intended for that antibiotic, they will create a target, which will mimic that target, but which will not cause, lysis or destruction of the bacteria. And then again, we have other mechanisms like they can be inactivated. We all know. There are enzymes that inactivates the antibiotic, and of these, sorry, there are three main enzymes which are responsible for drug resistance. One is the beta-lactamases, another is the aminoglycoside modifying enzymes, and the chloramphenicol acetyl transferases. These all destroy the antibiotics. But more important is the beta-lactamases. Again, in beta-lactamases, I'll just mention that there are different groups. But just please know that there are class A, class B, class C, class D, and all of this, which is not relevant. But then we say that many a times we say ESBL, extended spectrum beta-lactamases. These are the enzymes that hydrolyzes most of the cephalosporins as well. Whereas just the class A beta-lactamase, that is, for example, penicillinase, they conserve the cephalosporins. So whenever we think of antibiotic, what is important is whenever we plan to give antibiotic, it is important for us to think which is the most probable causative organism. Is it going to be gram positive or gram negative? Because the cell wall is different, and the drug which will act on the gram positive will differ from the 1 gram negative. And what is the site of the infection? And can it be a pathogen or can it be commensal? Because as I have mentioned, any bacteria which is isolated from our body, they may not be the pathogen. They are not always the pathogen. As I have mentioned, there are number of bacteria present in our body. And so is it a culture proven diagnosis or not? And which type of drug, which antibiotic is going to be effective for such organism? And what is the likelihood of developing resistance? And in the scenario, in the institute, or in that local area, what is the antibiogram pattern? What is the susceptibility pattern? What is the resistance pattern of that particular drug that you have in mind in the local region or in the institute? And in addition to this, what I would like to mention is about the PK and PD. PK-PD has become very important, particularly the PD. PD is important nowadays because we also when we say pharmacokinetics is the time and the concentration of the drug in the body. Whereas pharmacodynamics, it involves the concentration of the drug, okay, and the antimicrobial effect of that concentration at that particular site. So knowing the pharmacodynamics of that antibiotic is very, very relevant, because there are some which is not indicated by the pharmacokinetics. When we are dealing with antibiotic, these multi-drug resistant organism or drug resistant organisms, the role of pharmacodynamics is it's important. So let us not forget that the struggle of the mankind against infectious disease, it will continue and it will remain a major struggle. And what is the reason? What is the reason? Let us accept it that it is because of the imprudent uses of antibiotics in clinical practice. And as I have mentioned earlier, microbes are much more smarter. They are equipped with different mechanisms to develop resistance. So in India, it was published in Lancet in January 2011. In spite of the socio-economic progress, it's a little bit embarrassing. We should feel embarrassed that it was clearly mentioned that in spite of socio-economic progress, in India, and also because in spite of use of vaccines and antimicrobials in the past 60 years after independence from the colonial rule, infectious disease still contribute 30% of the disease burden in India. So this was clearly, antimicrobial resistance. This was clearly mentioned by Alexander Fleming. Alexander Fleming was awarded Nobel Prize for his discovery of penicillin, and he received the prize in 1940. And in his Nobel Prize acceptance speech, he has mentioned that the emergence of he has clearly mentioned everyone was so happy with the magic bullet of that time. But at that time in itself, Alexander Fleming has mentioned that there is a danger that the ignorant man may easily underdose himself, and by exposing his microbes to non-lethal quantities of the drug, make them resistant. That he had predicted in 1940. So each one of us here, we may think, why we are here? To be a steward in the fight against this drug resistance. So every health worker, whether it is a professor or a resident medical officer, or maybe a nursing superintendent, to a nursing staff, each and every one of us in the healthcare setup, we have a role to play. We may think that I can't do this alone. But then, please consider yourself, and I consider myself to be the tip of the spear. Because the tip of the spear, it decides the whole action of that spear. Okay. How it will go, what will be the depth, and at what angle if it goes, whether it will affect. So many are decided by the tip of the spear. And each one of us, we are the tip of the spear. Because we may not solve the entire problem, but remember, we can do a lot. So let us all use antibiotics judiciously, and let us all be involved in having an antibiotic policy, and let us make sure that I may not be it just can't be left to the one who prescribe it. Okay, the nurses have a bigger role. The nurses should be asking why these antibiotics, and the one who prescribe, the clinicians, they should always have an answer to that. And so the nurses, they should be empowered to ask why the antibiotics. Okay. So let us all be involved so that we conserve antibiotic, and the future generation, our children, so they say thank you to us. So thank you all for your patience here.